Bonsoir tout le monde. Everybody, good afternoon. Nous sommes réunis ici, le jury qui est devant vous, pour vous écouter, et visualiser et suivre votre présentation dans le cadre de la soutenance de votre thèse de doctorat en sciences portant sur la pectine issue des raclettes d'Opuntia Ficus India. Vous avez abordé le volet de caractérisation.
مرحبا يسونا كويس يعطيكم الصحة من دوسام دارت عاونا ما تبدميدنا. Mr. the President, examiners, honorable assistants, audience, I am very happy to present you the core of my work about the pectin from Opuntia Ficus indica cladodes, extraction, biochemical and structural characterization, and bioactivity. For introduction, polysaccharides are the main component of biomass, being the most abundant polymers available in nature. They are mostly extracted from renewable resources and well known for biodegradability and biocompatibility in such a way to achieve sustainable development. One of these well-known polysaccharides are pectin, with many and different application in agro and food industry and in biomedicine as well. But before application of pectin newly extracted in any domain, we have to answer such question about the eco-friendly extraction, their biochemical or chemical characterization, the structure and bioactivity. To understand my work, this slide is very important because it synthesizes all the whole work. So my work is divided in 4 steps. The first one is the extraction of the different pectic fraction. Then the optimization of only the most majority part, which is water-soluble pectin. Then the characterization of purified deprotonated major water-soluble pectin, and at last the cytotoxic or the bioactivity of this heat modified or heat treated major fraction. For our pectin was extracted from the cladodes or Opuntia Ficus indica. In our language we call it carmus. So, for the geographical distribution of the cactus, the term cactus from cactaceae refers to a group about approximately 1,600 species in 130 genera. The most important and most common and widespread is Opuntia genus. We consider that the early cactus family ancestor is from South American origin. It was introduced in southern Europe and Spain at the first expedition of Columbus at the New World. Thereafter, it was spread through the Mediterranean basin by the Spanish conquerors at the beginning of 6th century. Opuntia Ficus indica is the most important cactus species in agriculture today, and the first fruit crop especially adapted to semi-arid and non-irrigated lands. In fact, the Opuntia Ficus indica has a very special kind of photosynthesis. It is the opposite of the whole well-known photosynthesis in nature. Okay? So, for the other plants, the stomata are open by day and closed by night. But Opuntia Ficus indica are doing exactly the opposite way. The stomata are open are closed by day to prevent the evaporation, and open by night. In fact, the cactus save water through a special form of photosynthesis called CAM, or crassulacean acid metabolism. The cactus take in carbon dioxide at night when evaporation is less and store it in C4 molecules, which is the malonic acid, to use this molecule by day. The first step is the extraction or conventional extraction of the pectin and different fraction and the gelling behavior. The conventional method of extraction, we call conventional, so when we use the conventional method to heat the solution. So the three different fractions, water-soluble pectin and chelating-soluble pectin and acid-soluble pectin, were extracted from the fine powder of the Opuntia cladode with different conditions and different steps, but with the sequential method. The three extracted fractions were subjected to solid FTR analysis to confirm and to check the purity and to confirm their characteristics. It is clearly from we can see clearly from the spectra that they are generally similar, but they can be affected with the purity and the monosaccharide composition of each fraction. We can see clearly that the three spectra showed a well-resolved band at about 16-18 centimeter minus 1, which is characteristic of acid polysaccharide, which now has carboxy polysaccharide by opposition to the other class, neutral sugar. We checked to see the gelling behavior and the viscosity behavior of our whole pectin. I mean the three fractions combined. And we have seen that our pectin doesn't have and it doesn't show a well gelling behavior. So we tried to enhance this gelling behavior with copolymerization, with a well-known another natural polysaccharide, which is carrageenan. So we have seen that the less concentration of carrageenan, which gives us a blast augmentation and increase in gelling behavior, is 18%. Then we take this blend of 18% of carrageenan plus 82% of pectin, and we show in which condition they have the big viscosity. And we have seen the big viscosity at pH 2, as well as thermoreversible behavior, because we have a gelling behavior at low temperature, but solubilization at high temperature. We have seen also that our pectin is not sensitive to calcium bivalent cations. So we can say that the gelling behavior of our pectin is not the well-known egg box model. And the suggested model for the gelling behavior is the one with copolymerization and aggregation. For the monosaccharide composition, analyzed with high-performance ion exchange chromatography and the degree of methylation calculated with FTR, we have seen clearly from this table that the three pectic fractions are mostly composed of galacturonic acid, which is the building blocks of pectin molecules. The second table shows that the degree of methylation of the three pectic fraction as below 50. So we can say with international qualification that they belong to a low methylated pectin. And with the yield, for the yield, we've seen that the yield of WSP is 5.71, which is more greater than the two others. So henceforth, all the upcoming work will done only with WSP, and the two others are completely neglected. The second step is the optimization of microwave-assisted extraction of water-soluble pectin, the major fraction. For this reason, we have used a microwave home modified microwave. In fact, with microwave heating led to destructive changes in the plant tissues in such a way to increase the capillarity and the water absorption. With heating with microwave, there is a great pressure which builds up inside the material in such a way to break down the cell structure and better penetration of the extracting solvent. As a result, microwave-assisted extraction gives us an opportunity for a substantial increase in the yield of extractable pectin. The yield also depends on extraction operations as the temperature, the time, the pH, the ratio of solid liquid, etc. For microwave-assisted extraction, we have used a central composite design. Four factors with 3 levels were chosen to explore and optimize the influence of the process variables on the maximum pectin extraction. The first factor is the solid-liquid ratio. The second is the pH. The third is the extraction time. The fourth is the microwave power. And this diagram is the sequential step to use it to extract the WSP, water-soluble pectin, with using different combination of microwave, time, pH, and ratio. The extraction yield of pectin Y is selected as a response. The results were fitted into empirical second-order polynomial model as shown in the following equation. So this one is the equation of the second second polynomial model of our experimental design. Okay. The GMP software used to construct the 3-dimensional response surface give us this matrix with 29 different experiments representing the different combination of the factor x1, x2, x3, x4. Then we have in this column the experimental yield, and in the other we have the predicted yield given with the software. The regression analysis gives us a R square about 0.97, which is very high, and that means that only 3% of the variation are not explained with our model. Then we can see with ANOVA analysis that the R square and adjusted R square are very close. For a good statistical model, R square should be close to R square, confirming that the model is highly significant. In other hand, we have R square adjusted and R square were reasonably close to 1, 0.97 and 0.93. So, which indicates that the high degree of correlation and between the observed and predicted values. In other hand, we have a great F value with a small p value indicates that the model is significant and can be used to optimize the extraction variables. This one is an example of the 6 different surface response obtained. The optimum condition of microwave-assisted extraction with are follow. The time is 2 pounds 15 minutes. The microwave is 518 watt. The pH was 2 pounds 26, and the ratio was 1 gram per 15 milliliters. Under this optimal condition, the model predicted a maximum yield response of 12.56. This optimum condition used in an extra essay to check the validity of our model, and the obtained yield was 12.7. It is a bit greater than the predicted one, but with the test student there is no significant difference. So we can say, finally, that this result confirming that the model was adequated for reflecting the expected optimization. Now, to check if the microwave treatment doesn't have any alteration of our pectin, we have analyzed pectin extracted with different treatment with microwave, 200, 400, and 600, compared with the conventional method used in commercial condition. And the spectra are very similar, and the distribution of functional group is not altered. So we can give a conclusion that microwave treatment doesn't affect the pectin structure anyway. The third step is biochemical and structural characterization. For analyzing the molecular weight distribution, we have analyzed our pectin with high precision size steric exclusion chromatography. Both absorbance and refractive index signal were acquired. The absorbance signal in red was superimposed with refractive index in black. The two chemical class calibration standard used, which are purulence and dextrans. And the two columns coupled in series, we have used this condition in such a way to achieve, to encompass a broad range of molecular weight distribution. We can see clearly from the spectrum that our pectin is mostly composed of two kinds of pectin. We have the high molecular weight and the low molecular weight. And the strong peak at 218 to check the 18 nanometer to check the protein content is very high and related to the fraction eluted between 18 and 20. The minor, and you can see that the minor protein content is highly connected to the low molecular weight fraction because of the next slide, please. The deproteination step with Sievig method. We have made three different step deproteination. The absorption peak at 218 is decreased but still present. So we can't give a conclusion that the pectin is highly reduced, but still very connected to low molecular weight pectin. In fact, the protein in native WSP is 0.66%. And the deproteination extent is about 90%. You go on? Now, to check the size and the conformation of our pectin, we have analyzed our pectin with dynamic light scattering and static light scattering. In fact, for the dynamic light scattering, please, you can give all the slides. Okay? No. So for the dynamic light scattering employs the measurement, the dynamic light scattering, this one, gives the measurement of the scattered intensity at very short intervals of time. The fluctuations in the intensity have a characteristic fluctuation time that reflects the diffusion coefficient of the molecule, which will now in biophysics as Brownian motion. The hydrodynamic radius of our particle pectin is the radius of the spherical particle with the same diffusion coefficient. This one is the analysis with dynamic light scattering. Now the analysis with the static light scattering, it employs the measurement of the scattered intensity at several scattering angles. For dynamic light scattering, we use just one angle, it's 90 degrees, but the static light scattering, we use different angles. The light scattered from the solution macromolecules has an average intensity that reflects the molecular weight of the molecule. The mass duration radius, Rg, was determined also. You go on? This one is the interpretation of dynamic light scattering result as electric field correlation function at 90 degrees scattering angle for the solution of WSP using the Einstein-Scott's equation. We calculated the hydrodynamic radius. It is about 110 nanometers. And the result of the scattering angle, which are not presented in my thesis because the results are not published yet, but the result as given here just to have an idea about what is the size. So the result of static light scattering as represented as a Zim plot. This one is the Zim plot. And the molecular weight was calculated as extrapolation at C0 concentration and 0 angle. So the molecular weight calculated was about 10 power 7 gram per mole, which is very huge. But most important is the radius of gyration. It is about 325 nanometers. If you remember, we have I said that the hydrodynamic dynamic radius is 110. So the duration radius is greater than hydrodynamic radius. So we can have a conclusion that our pectin is not spherical, but is more elongated. You go on? Now, the structural characterization or the chemical characterization of our pectin. For this reason, we have used three different chemical derivatization. The first is the neutral monosaccharide composition detected as alditol acetate derivatives. The second one is the ring 4 and anomaly, which now has absolute configuration. They are analyzed as trimethylsilyl derivatives. And the last one is the linkage pattern analysis as with permethylation and methanolysis. For the first one, alditol acetate prepared with the first step of hydrolysis, then reduction and acetylation to have alditol acetate derivatives. Okay. Next one. We have detected 5 different alditol acetate. The first is galactitol from galactose. The second is sorbitol from glucose. The third is xylitol from xylose. And the fourth is rhamnitol from namnose. And at last, we have arabitol. But in this case, it is not from arabinose, but it is from the decarboxylation of galacturonic acid, as it will be proved further. Okay? For the trimethylsilyl derivatives, we have the first step is the permethylation, then the methanolysis, then we have the trimethylsilylation. Okay? With this method, we have detected about 15 different cyclic forms. But the most important as the galacturonic, as galactose with beta and alpha anomers with the pyranose form, then the galacturonic acid with as a decarboxylated form, and finally we have the glucose. Okay, the next. The partially methylated alditol acetate, which is the method used to check the nature of the bound or the linkage pattern. We use the first step of permethylation, then a second is hydrolysis, and finally we have reduction and acetylation. Okay? And we have 6 different linkage determined. Galactose linked with 1,6 position. The second is glucose at position 1,4. The third is rhamnose at position 1, 3, and 4. And at the fourth, we have terminal glycose. And we have glucose at position 1 and 6. And finally, we have a terminal xylose. You can go before. If you have seen here, if you remember that, I have said that the most important building blocks of our pectin is galacturonic acid. But it is not. The linkage of galacturonic acid is not detected here for the simple reason that galacturonic acid is degraded under the reduction condition of this method. Okay? So we need another supplementary method to check only the galacturonic acid linkage. And this method is called, you go on, is called carboxyl reduction method. In fact, this method aims to transform the galacturonic acid to its corresponding neutral sugar. So instead to detect galacturonic acid, we will detect galactopyranose, the galactose, okay? And the analysis with carboxyl reduction, we have detected a peak, you go on, the detection of digalactitol 1,4,5,3-acetate and methylated at the position 2, 3, and 6. But the most important, it is deuterated at the position 6. It confirms that our galacturonic acid has a specific linkage of 1,4. Okay? Now the degree of methylation of our pectin, WSP, after deprotonation was checked with FTR. For this reason, we have used different commercial pectic fraction with a known degree of methylation. From the calibration curve, we have calculated the degree of methylation as 8.46. You go on? Now, to give a proposal of our hypothetical structure of our pectin, we have to use the data from alditol acetate, TMS, partially methylated alditol acetate, the carboxyl reduction, the standard used, and the FTR and degree of methylation. We have to use all this data, and the given proposal and hypothetical structure of our pectin is this one. It is composed of the principal backbone of rhamnogalacturonic, so it is a rhamnogalacturonan with a slight chain connection on the rhamnosyl residue. The last step is the bioactivity as antioxidant and cytotoxic activity. Okay, you go on. For the antioxidant activity was checked for the WSP. We have checked two different antioxidant activity, the free radical scavenging activity and chelating activity. For the cytotoxic activity, we have used viability test as MTS and trypan blue, the Hutch nuclear staining to check for the apoptosis and the cell program death, and finally the propidium and annexin 5 nuclear staining to make the differentiation between the necrosis and apoptosis. Okay? This one is the result of scavenging activity. You go on? We can see clearly that our pectin has a huge, a very important scavenging activity of both hydroxyl radical and DPPH radical comparatively to the standard one, the ascorbic acid. You go on? As well as it have a great chelating activity of ferrous bivalent cation comparing to the commercial chelating agent as DTA. You go on. The MTS test, in this test, the metabolic activity of the cell can be assayed as an indication of cell viability. The EC, based on the capacity of mitochondrial dehydrogenase enzyme in a living cell, it's very important, in living cell, to convert a yellow soluble tetrazolium salt to a dark formazan product. Okay? So for the MTS, solution was given from Progema, applied directly on microplate on the cell, and the the lecture was done at 49 nanometers. The result on the cell viability with MTS test and carried out with heat-treated, deprotonated water-soluble pectin as here. And we see clearly that our heat-modified pectin is affecting selectively the LAN5 cells without any effect on the NIT333 cells. In fact, the LAN5 cell are the cancer cell derived from neuroblastoma. And the NIT-T33 are the normal cell of fibroblast. You go on? Go on? The trypan blue exclusion test is a complementary test used. It is more helpful to stain the cell with a day, such a trypan blue. In this case, viability is measured as ability of cell with a healthy and compromised membrane integrity to exclude the day. This one is the structure of trypan blue, and the result expressed as, not as before as viability percent, but as cell death, confirming the augmentation of increase of death with increasing concentration time of exposition to heat-treated pectin. Okay? For the Hutch nuclear staining, this test staining have been performed in order to confirm cell death with the detection of apoptotic bodies. The molecule of Hutch is staining the DNA molecule with fixation on the small groove, and with excitation with UV radiation, it gives a bright blue coloration. And here, with the treated sample, we can see the formation of small body. A small body, which is the chromatin and DNA fragmented. We can call in physiology as apoptotic body. But in the other one, we cannot see the apoptotic body, and this test reveals that the apoptosis really happens under treatment with modified pectin. And finally, we have to discriminate between the early and late apoptotic, and between necrosis, we have used the staining and labeling with propidium iodide and annexin 5. In fact, the annexin is a small molecule which have a high affinity for phosphatidylserine. And phosphatidylserine is a phospholipid. In the normal condition of the cell, it is presented in the inner part of the plasmic membrane. But in the apoptotic condition, this phosphatidylserine undergoes a flip-flopping externalization to the outer membrane, and it presents a signal of eat me, manger moi, eat me signal. Okay? It is a call for phagocytosis. And annexin in this way, when the phosphatidylserine has externalized, it can bend and give a signal. So we have three different cases here. For the living cell, it is the phosphatidylserine is inside, so it cannot be stained, and the propidium iodide cannot penetrate. So the healthy cell, the normal cell, is not colored. For the early apoptotic, we have a beginning of externalization of phosphatidylserine, so it will be only stained with annexin. But the late apoptotic or necrotic cell, we have degradation of the plasmic membrane, so the propidium go inside the cell, and with the externalization of phosphatidylserine, it will be labeled with both propidium and annexin 5, and we have the three different coloration, with the simple treated with modified pectin, and this one is the control which is not treated. And we can see the difference between the both, that our pectin induce cell cytotoxicity of the cell. of the cancer cell, of course. Len 5. You go on. As conclusions and perspectives, Opuntia Ficus indica can be considered as a valuable renewable resource for green extraction of pectin. Pectin without any modification, chemical modification, is very promising for biochemical application and food industry perspectives. And the pectin showed a very promising free radical and chelating capacity, and we have the heat modified pectin cytotoxic activity is affecting selectively the cancer cells without any effect on the normal cell. It's very important. So we can use this pectin for microencapsulation as well as for drug delivery system. And finally, the result in our results are in line with many other previous works, and further assessment of molecular mechanism of cytotoxic properties should be a significant contribution for therapeutic purposes. Our work have given birth to 4 different articles in highly impacted journal. The first one is International Journal of Biological Macromolecule. The second is Food Chemistry. The third is another one in International Journal of Biological Macromolecule. And at last, we have Industrial Crops and Products. And we have done 3 different international oral conference proceedings. My thanks for my Professor Khadir Madani for his friendship, for his human qualities, and for his proficiency. And my warm greeting for all the BBBS team. And finally, my special thanks for my team at the Institute Biophysique in CNR, Palermo, Italy, under the direction of Dr. Giacomazza Daniela. And the special thanks for my close friend and office mate in Palermo, Samuel Racusta and Loredana Randazzo. Thank you.